In this lecture, we will discuss about some general terms in the biology, or you can say that bio-related field, so that it will be easier for us to understand the further lectures. So let us start with the wild type. Let us start with the wild type. What do you mean by wild type? If someone reads any paper, and most of the paper that is related to biology has this term wild type. The wild type means that things that exist in nature as it is. For example, cells without any treatment. And knockout. Knockout can be described in different aspect. One of this aspect is gene knockout. So what is gene knockout? Gene knockout means that in those kind of cell, that gene is made inactive by any techniques, by any of the technique that is available, or through some new techniques or some novel techniques. So one of some of the examples to knock out the gene is CRISPR. Technology. CRISPR is the latest thing to knock out gene. Other things to knock out, other technology to knock out genes are too much lengthy, and most of the time now people uses CRISPR to knock out any gene and then analyze the behavior of the cell. So knockout means to make inactive any inactive any particular thing such as gene. That gene can be responsible for producing a specific kind of protein, and the experiment was about to know the impact of this gene, or to confirm that this gene does that function. So what people does is they knock out this gene or inactive this gene, and then see whether that function is still been occurred in the cell or not. For example, in a wild type cell, we have as we have learned in one of the previous video, that calnexin responsible for proper folding of the proteins. So if we knock out calnexin from that cells, then we should be able to see some proteins that are not been properly folded, or some proteins are even not been shown up in that calnexin knockout cell, because they are so improperly folded in that the endoplasmic reticulum ERAD system, that is endoplasmic reticulum associated degradation, detects that non-native structure and directs them towards degradation. So this kind of knockout studies are being done to confirm the function of a particular protein of interest or gene of interest. And then is the expression data. Expression data is link to the previous two points. Expression data, what previously earlier when the biofield is emerging, expression data is expression data. Suppose we have in order to compare the expression data, what are the problems that people are facing earlier is that suppose there is a library that has expression data of all the organs of human cells, and someone has done an experiment on just a liver cell, and if they compare the expression data in this, if they use this library of all organ cells and compare their experimental data, they will get the output in terms that their data is more expressed towards liver. However, this is what the experimental already knows. So they need to understand what are the things that are upregulated in the liver. So in that case, instead of using this library, all organ library, they should have used healthy person, liver versus affected person, liver sample. In that case, people will be able to get a more in-depth idea about what are the things that is going on in the affected person with respect to the liver. So in this case, the healthy liver is data is considered as the wild type, and this affected person liver data is considered as the sample of interest. So it might be possible that in the affected person some gene might have altered or knocked out, but we don't know about it. That will be known by analyzing the data and using some of the bioinformatic tools to come to any conclusion. So, and what are the general criteria that usually people used to consider that the thing is significantly upregulated or downregulated? General criteria that people use is log 2 fold 1.5 G. At least they need log 2 fold 1.5 is equal to point. I don't know exactly. Let me do it more easier. If something is expressed 1.5 times higher or lower than wild type, then it is considered as upregulated or downregulated. And there are many other ways to confirm this, many statistical tools to confirm this, such as p-value test or p-value test. We need to take care of the multiple hypothesis also. That we will discuss it in the next video. For this, we just need to understand that if we think we need to understand the general criteria is if something is something is 1.5 times more or less than the wild type in the sample of interest, then that is considered as that is not considered as the normal behavior. That protein is either affected. That protein is affected by something in the sample of interest. Okay, that's all for this video. Thank you.